Erika Garcia, and she'll be talking about the examination of Chelicerae shape, morphology, in camel spiders using elliptical Fourier analysis.
All right. So thank you for everyone for joining in today. Like I said, my name is Erika Garcia. I'm a doctoral student at the University of Colorado, Denver, and at the Denver Museum of Nature and Science. And today I will be presenting a portion of my doctoral research regarding male cheliceral morphology and the elliptical Fourier analysis. So I'll first start off by saying that there are two North American solifuge families. One of them is the Eremobatidae, which will be the focus of this talk today. And their range is from southern Canada down to southern Mexico. And currently there are two subfamilies within this group. There are the Therobatinae and the Eremobatidae. There are 8 genera, 18 species groups, and 187 species described, making them one of the most diverse solifuge families. So the arachnid order Solifuge are recognized by their robust, powerful jaws, or their Chelicerae, and these Chelicerae functionally serve both sexes for a variety of purposes, one of them being fighting. So these are two Eremobatidae males fighting with each other. So they use both their pedipalps and their Chelicerae to fight each other off. And this video is slowed to 25x. This is really fast, this footage. And another purpose is for burrowing. So this is footage of a female Eremobatidae constructing her burrow. So she uses her Chelicerae to move dirt out of her burrow during burrow construction. And this video was also slowed down. And of course, they use their powerful Chelicerae for feeding. So this is an Eremobatidae male pulverizing a fly. So they pulverize their prey with their Chelicerae. So despite this overlapping utility in both sexes, cheliceral morphology is sexually dimorphic. So this is an example of one species of Eremochelys. We have the male on the left and the female on the right. So there's a high degree of morphological variation within the males, and they vary across taxonomic levels. So they vary strikingly from different genera to species, and they also vary intraspecifically. And some of those variable characters that are within the cheliceral morphology are dentition, the shape of the manus, so this structure here, the shape of the fixed finger, the shape of the movable finger, and also the shape or the presence absence of this character here, this spondyle notch. So despite this high degree of variability within the males, females exhibit a high degree of morphological conservatism. However, dentition may be useful in species identification, and this is an area of ongoing research. So given this high degree of morphological variation within the males, this variation inspired much of the groundwork of Eremobatidae taxonomy. So here's an example of the striking morphological differences that occur between the different genera of Eremobatidae. And this variability can be attributed to their role in sexual reproduction. So here's a quick clip of Eremobatidae mating ritual. So here on the right, we have the female. Here on the left, we have the female. On the right, we have the male. And what the male is doing is he's using his Chelicerae to deposit his sperm packet into the female genital opening. So this morphological variability is likely due to sexual selection pressures. However, those exact sexual selection pressures driving this morphological change is currently unknown. So in 2015, the first and only available family level phylogeny was published for the family Eremobatidae using a multi-locus data set. And I do want to point out some groupings here. So first we have the Therobatinae. So within this box is most of the Therobatine genera. There are 4 genera within this subfamily. And this other grouping is the other subfamily, the Eremobatidae. But one thing to point out is that within the Therobatinae, this group, many of the genera are polyphyletic. So Hemerotreca and Eremochelys are both polyphyletic genera. So given this result, this suggests that the Therobatinae primarily based on male chelicerae morphology, is in need of taxonomic revision. So as a smaller segment of a larger monographic revision and as part of my doctoral work, this research that I will be presenting on today involves a re-examination of male chelicerae morphology using 2D cheliceral outlines and morphometrics, as well as model-based clustering algorithms to objectively determine distinct morphological groups within the Therobatinae. So to conduct an outline analysis, of course, we have to generate outlines. So how I generated these outlines was I dissected the right chelicera from multiple Therobatine representatives in the Denver Museum Natural History collections. And I generated multiple images using a Leica stereo microscope, and using the stereo microscope, I took images of the retrolateral views of male chelicera in the Leica software, and binary images, so black and white images, were generated using this free photo editing software called GIMP. So to analyze outline shapes, I implemented this method. So this is the elliptical Fourier analysis. It was introduced by Kuhl and Giordana in 1982. And this method was developed to quantify shape outline. So it quantifies shape outline using something called the Fourier series. This Fourier series was developed by Joseph Fourier. And this is a quick example of how it works. So the Fourier involves simple harmonic motions. This is one simple harmonic motion. So this is a periodic function of an oscillating sine wave. And this oscillating sine wave moves at a constant frequency. But when you change that frequency, you can generate different types of sine waves. So a fine scale sine wave or a coarse scale sine wave. And the way that Fourier series is generated is you take multiple harmonics. So each of these harmonics are added together to create the Fourier series, and the Fourier series is able to generate a completely new sine wave by simply adding each of these sine waves together. So the Fourier series can characterize any outlines that are superimposed on an X and Y coordinate plane. So here are the two equations involved in the Fourier series. So this equation up here classifies all of the coordinates in all of the X coordinates, and then this equation here classifies all of the Y coordinates. And it's a series of additive cosine and sine functions to describe the overall shape per harmonic. And the Fourier series is quite remarkable because it can draw nearly any complex shape. So here's a simple diagram of how this works. So by iteratively adding different harmonics, we're able to summarize the shape outline. So here's another example in real time of how that Fourier series works. So each of these circles represents one harmonic, and these harmonics are interrelated, and these harmonics move collectively together to characterize each of these data points, so each of these X and Y coordinates. And by simply changing the size and the angle of each vector within these harmonics, it allows for flexibility in drawing nearly any shape. So this serves for a lot of purposes, and there's a lot of benefits to this method, such that there's no need for determining homologous landmarks as in other morphometric methods. And it considers overall shape. So it considers all the subtleties and details of the shape outline. So I implemented the elliptical Fourier analysis in the software package Momox in R. So I imported all of my outline images into R, and I performed a series of smoothing iterations. So here's an example of how smoothing works. So within
10 minutes. Yeah.
So thank you. With an increasing number of smoothing iterations, you kind of lose a lot of detail. So I limited the number of smoothing iterations to less than 50. perform 1,000 interpolations. And then due to my interest in considering overall form, I rescaled each of these images to depict actual size. And then the number of harmonics were determined and Fourier coefficients were calculated. So those Fourier coefficients can be summarized in these equations here. So there are 4 coefficients per harmonic. So this is a simple data set. So this is a mock data set. So if there was 2 harmonics calculated for this data set. So these harmonic coefficients are used to reconstruct shape, and they are used in downstream analyses. So I performed a principal components analysis, and I isolated the first 3 principal components for downstream analysis, and I implemented 2 model-based clustering algorithms. These are unsupervised machine learning algorithms to determine the optimal number of clusters in my data set. So here are the results of the PCA. So this is using a smaller data set. So this is with 18 representatives of Eremobatidae species. So some patterns are able to be deciphered in morphospace where size matters. And there's also 2 primary characters that are important in distinguishing some of these morpho groups. And that is the dentition on the movable finger and the presence or absence of this fondal notch. So here's another example of the principal components analysis using an expanded data set with 38 species. And this is showing the current taxonomic boundaries as such. So moving on to the clustering algorithm. So with a data set of 131 representatives, there was strong support for 5 clusters, and each of these cheliceral shapes represent the mean shape for each cluster. And some notable characters are the shape of the fixed finger, the distinctiveness of the fondal notch, and the dentition on both the fixed finger and the movable finger. Similar results were recovered using the Chelicerae process mixture modeling as the Bayesian clustering algorithm and this analysis also recovered similar patterns as the Gaussian mixture model. So in conclusion, there are several informative characters that were teased apart using the PCA analysis, and that was the presence absence of the fondal notch, and the shape of the fondal notch, dentition, and the length of the Chelicerae. And the model-based clustering algorithms support 5 clusters within a data set of 131, showing support for patterns in these character sets, such as the fixed finger and movable finger dentition, as well as the shape of the fondal notch and the shape of the fixed finger. So this approach, using both the model-based clustering algorithms and the elliptical Fourier analysis, shows that there's promise even at the generic level. So with that, I'd like to thank these folks for their helpful discussions, our NSF grant, Paula's NSF grant, and the American Neurological Society for hosting and organizing this meeting.
Great, thank you so much. Lots of really great comments in the chat, mostly talking about especially your description of the process. Jeff Schultz though asks, how do you control for differing position of the movable finger against the rest of the Chelicerae?
Yeah, so when generating the images, I try to standardize the images because the orientation of that movable finger does matter. So I try to either position it when taking the images, or I manipulate it in the photo editing software so that they're all in the same position.
Very good. Sebastian asks, have you noticed any trends besides species ID in the morpho clusters, such as ecology or courtship or so forth?
I haven't examined those, that area. That's something that I'm interested in further down the line. So I'm interested in implementing ancestral state reconstruction once we get a solid UC phylogeny for this group. So yeah, I'm not sure about that yet.
Okay, thank you so much. That's actually all the time we have for questions. So there's a couple more in the chat though for you to address.